Hello everyone and welcome back to my channel. In today's video, we shall discuss about the structure of enzymes. Now, enzymes is a topic that we all are introduced to in our mid school, and we barely scratch the surface of enzymes in high school. We are introduced to its structure and how enzymes are classified and what are the different kinds of enzymes and what is their basic function. But if you're pursuing your higher studies, if you're a student of biochemistry, then it is of great importance to understand the concept of enzymes. So, what enzymes are actually? Enzymes are biomolecules of very high molecular weight which catalyze biochemical reactions. Almost all biochemical reactions that we know of today, it could be anything from digesting a bowl of ramen noodles to flexing your muscles in the mirror. Almost all biochemical reactions are catalyzed by enzymes. In fact, there is a study which says that if there were no enzymes present in our body, a bowl of these ramen noodles for dinner would actually take months and months to be fully digested. So, with the presence of enzymes, such biochemical reactions are hastened. Okay, they are sped up. The speed is increased. The rate of these reactions is enhanced. So enzymes, they function to catalyze, they function to enhance the rate of the reaction, increase the speed of the reaction. So here we begin with the lecture. Almost all metabolic processes in the cell need enzyme catalysis in order to occur at rates fast enough to sustain life. An enzyme, like I said, is a biomolecule, either a protein or a ribonucleic acid. It could either be a protein or a ribonucleoprotein that catalyzes specific chemical reaction and does not affect the equilibrium of the catalyzed reaction. So here we have a lot of terms to deal with. Firstly, an enzyme could either be a protein or an RNA. If it is a ribonucleoprotein, then we specifically call it a ribozyme. If it is a ribonucleoprotein, we specifically call it a ribozyme. Example would be peptidyl transferase, which catalyzes the formation of peptide bond. Peptidyl transferase. Okay. And these enzymes catalyze only specific chemical reactions. Yes, this is a characteristic of enzymes that enzymes are very specific to the substrate that they bind, very specific to the reaction that they catalyze. Okay, and it does not affect the equilibrium of the catalyzed reaction. Does not affect the equilibrium means it does not change the direction of the reaction. It does not change the steady state of the reaction, which means that the delta G remains unchanged. Now, from your knowledge of physics, delta G is signifies delta G signifies Gibbs free energy from thermodynamics. Now, to understand what basically is Gibbs free energy, just it's a very simple concept. Imagine Gibbs free energy is actually, in simple terms, the total available energy in the system. All right. So, if we consider Gibbs free energy as the total energy of the system, so think that you have organized a party at home. Okay, and the total energy of the party could either be the total energy, the whole enthusiasm and excitement of the party could either be in negative terms, that means negative del G, or it could be in positive terms. If the party, that means the delta G, okay, is in negative terms, that means the party is happening on its own. There is no extra effort that you need to put in. You do not need to call a DJ or provide an external source of entertainment. You do not need to organize any games, nothing like that. People are just having fun, they are talking amongst each other and getting excited, and the party is spontaneously happening on its own. The party is spontaneously happening on its own. You do not need to put in extra effort. But if the value of delta G is positive, that means the available energy in the system, you need to supplement it. You need to facilitate the whole energy with an external source of energy that could be in the form of heat or light. Okay, so to make the party happen, to make the reaction happen, you need to supply energy in the form of a DJ or some external source of entertainment is needed, so that the people, they interact amongst themselves. People over here, I mean the molecules, they interact amongst themselves, right? So in case of enzymes, the last line says it all, that the last line says it all that enzymes catalyze only thermodynamically favorable reactions. So if the delta G, the value of delta G is a negative, that means these reactions are happening on its own, spontaneously happening on its own. The problem is that without an enzyme, the reaction would take much, much, much longer to be fruitful, to give us the product. It would take much, much, much longer. Okay, but with the help of enzymes, what happens is this time is shortened, the speed of the reaction is hastened. Okay, the reaction happens at a rate fast enough, it enhances the rate of the reaction. And how does this basically happen? By decreasing the entropy of the complex that is formed between the reactant and the product. By decreasing the entropy, entropy in thermodynamics is denoted by the letter S. So by it happens by decreasing the entropy. Entropy in simple words means randomness. In simple words, it means randomness. So imagine that a classroom without a teacher. Then what would happen? The students are talking amongst themselves. There is chaos. They are not in their place. They are roaming everywhere. Right. So the randomness of the classroom, the system is very high. But once the teacher enters the classroom, an order is maintained. Children are put in the correct order. They are put in to sit in the correct seat and made to study, right? That means made to give fruitful result, right? So enzymes do the same task. They increase the rate of the reaction by decreasing this randomness of the whole system, and they do this by forming a transient complex with the substrate. Over, what do we mean by a transient complex? Transient means it is just temporary. It is not a permanent complex that is formed between the substrate and the product. Substrate and the enzyme. It is completely a transient complex that is formed, and we can denote this by writing enzyme with the substrate. It forms a transient complex. Okay, and this es complex will form the product, and as we know that the enzyme will be released. Okay, so this es is a transient complex with the substrate, and they do this by providing a lower activation energy. Very important, they do this by providing a lower activation energy. What do we understand by the term activation energy? Activation energy is nothing but the energy barrier. So you can simply understand this by relating activation energy to the pass percentage required in your examination. That is the minimum bar. It is the barrier that you need to cross to give a fruitful result. That means the product. You need to cross that barrier to give the fruitful product, right? So they do this by lowering the activation energy. Obviously, if the pass percentage is lowered, supposedly if it is 40% in your institute, and if it is lowered down to 20%, it is easier for you to qualify for the examination, right? So enzymes do the same thing. They lower the energy required for to release the product. They lower the activation energy such the reaction is more favorable, it is enhanced. I hope I am clear. If not, you can please leave your comments down below, your questions and suggestions. So over here, what I have represented on screen is a graphical representation of how enzymes function. On the y-axis, what is denoted is the free energy. That means the delta G is denoted, the free energy, or rather the available energy in the system. Okay, now the value energy. And on the x-axis is the progress of the reaction. Without an enzyme, the blue line, if you see clearly, the blue line without an enzyme, the energy of activation is much higher. The energy of activation is much higher. So I can say this is 40% marks that are required for you to qualify for the exam. Okay, but with the help of an enzyme, this barrier is lowered 20%. But the path of the reaction, as you can see, this path of the reaction remains the same. Only what is happening is that enzymes are facilitating to achieve this path. They are facilitating the reaction. They are enhancing the rate of the reaction. They are enhancing the rate of the reaction by increasing the speed of the reaction. Okay, and in the case of the reaction with an enzyme, the delta G, that means the total energy of the system, does not change. The total energy of the system, no matter you need 40 marks to qualify the exam, or you have lowered the bar to 20% to qualify for the exam, the syllabus remains the same. What you have to study remains the same. What energy is required by the system, that remains the same. That means delta G remains the same. Only the fact is that with the enzyme, with the help of enzyme, the attainment of the product, the attainment of this energy becomes much easier. Okay, a significant part of the used for enzymatic rate enhancement is derived from weak interactions. Once again, let me remind you that enzyme and the substrate, they form a temporary bond. Okay, it is a transient complex because the enzyme has to change the substrate. It has to form conformational changes into the substrate to give us the product. So this cannot be a fixed bonding, it cannot be a permanent bond. And that is why there are only weak interactions between the enzyme and the substrate. Okay, what kind of weak interactions are these? Like hydrogen bonds, hydrophobic interactions, ionic interactions between the enzyme and the substrate. The enzyme's active site is structured. The enzyme's site is structured to ensure proximity and orientation of the active site with the substrate. So on your screen, you can see a GIF. This GIF shows you the enzyme, which is brown in color, and you can see the molecules of the substrate, which are green in color, entering the enzyme. But they are not entering the enzyme everywhere. Okay? They are not binding with the enzyme from here. It is not binding the enzyme from here or here. There is a particular site, a particular position where the substrate is binding, right? And that particular position, this one, is called the active site. The site of the enzyme, the particular position of the enzyme, the particular place on the enzyme where the substrate has to bind, that is called the active site of the enzyme. And that active site is structured specific to every enzyme. to ensure the perfect proximity and orientation with the substrate. This can be understood by the concept of lock and key, and actually there is a lock and key hypothesis also which you will study. Now every lock has a specific key. So here you can imagine the lock to be the enzyme and the key to be the substrate. So every lock, that means every enzyme has its specific key, which is the substrate, right? And once you enter the key into the keyhole, that with the keyhole acts as the active site. That active site, the keyhole, has a specific orientation, has a specific design which matches with the particular key only, right? The keyhole has a specific design to match the conformation, the orientation of its particular key. Not every key can fit into every lock, and this is because of the orientation, the whole configuration of the keyhole specific to the lock. I hope I am clear. Okay. So there are a lot of these multiple interactions which need to happen between the enzyme and the substrate, and exactly where do these interactions have to happen? At the active site. So that is why enzymes actually have a very large size. All right. The binding energy can be used to lower the substrate entropy. Once again, the substrate randomness. The binding energy can be used to lower the randomness of the substrate of the whole system, or to cause a conformational change in the enzyme, which is called induced fit. Obviously, you are inducing the fit, the conformation between the substrate and the enzyme. Imagine when we insert the key into the lock, does the lock open on its own? No. You need to push it. You need to induce the fit between the lock and the key. You need to twist the key, right? So there is an induced fit. And in the system of enzyme and substrate in biochemistry, what is the factor which is inducing this fit? कौन कर रहा है ये conformational change? ये induce कौन कर रहा है? It is the binding energy. It is the binding energy which is inducing this fit, this conformation between the enzyme and the substrate. Binding energy accounts for the exquisite specificity of the enzyme for the substrate. The specificity is because of this conformation, the whole shape and structure of the active site specific to the substrate. Okay. So as you can see over here, the substrates that begin with the particular energy in the system, okay, facilitated by the binding energy, catalysis occurs, they reach a transition state, and then the products are rendered. Okay, so what is happening? The catalysis is happening because of the energy that is supplied by the binding energy between the enzyme and the substrate. Unit of enzyme activity, the international unit of enzyme activity, it is defined as the amount of enzyme causing a transformation of 1 micromole of the substrate per minute. 1 micromole of the substrate that is transformed into the product per minute at 25 degrees Celsius under optimal conditions. Okay, so one international unit activity, 1 EU, is equal to 1 micromole of the substrate transformed into the product per minute. Another essential unit of enzyme activity is catal. 1 catal is defined as 1 mole of the substrate transformed per second. 1 mole of the substrate transformed into the product. 1 mole of the substrate per second. In each second, how many moles of the substrate are transforming into the product? Specific activity is what is defined as micromoles of the substrate transformed per minute. Molecular activity is defined as micromoles of the substrate transformed per minute per micromole of the enzyme. So on your screen, you can see a chart where the rate of the reaction, how were the units of the rate of the reaction are given, micromoles per minute. Micromole is the substrate, the quantity of the substrate transformed into the product. Enzyme activity is the unit is micromoles per minute. Specific enzyme activity micromoles per milligram per minute. Okay, so this is how the units are used. So once again, what are enzymes? That is what we are here to discuss. All known enzymes are proteins, but they could also be ribonucleoproteins, which are known as ribozymes. These are high molecular weight compounds. Why are these high molecular weight compounds? Because they have to have a very crucial active site and perform certain conformational changes between the substrate and the enzyme, performs, so many interactions need to be facilitated, so it needs a very large area, and these enzymes are of high molecular weight, principally made up of amino acids. Obviously agar wo proteins ka hai to it is made up of amino acids, all joined by peptide bonds. Getting deeper into the structure of enzymes, enzyme structure can be studied under two headings: simple and compound. When we talk about simple enzymes means that we are only talking about the protein component of the enzyme. We are only talking about the supposedly this is an enzyme. We are only talking about the proteinaceous component of the enzyme. An enzyme in an inactive form. An enzyme in an inactive form is called a zymogen. Example, chymotrypsinogen. Whereas an enzyme, if it is if it is in active form, then it is called an apoenzyme. So it would be right if I said, sorry. So if it would be right if I said that if this is a functional enzyme, I could call this an apoenzyme, right? Example of apoenzymes are trypsin and pepsin. Trypsin ka hi inactive form is chymotrypsinogen. Okay. Talking about conjugate enzymes. So conjugate enzymes, that means you will have the main protein component of the enzyme, the proteinaceous enzyme, functional enzyme. So I will call this the apoenzyme. Okay. Along with a non-protein component. So apoenzyme along with the non-protein component is called a holoenzyme. That non-protein component could either be inorganic or it could also be organic. If it is an inorganic metal ion, if it is an inorganic metal ion, supposingly in in its structure you are having a metal ion, say nickel, or you are having ferric ion. Okay, there are many examples which I will show you. So this is the region of the inorganic metal ion, supposingly nickel. Then this structure is called a cofactor. The apoenzyme in A, the functional enzyme in A, red A, along with the blue marked cofactor, both of these things together are now called a holoenzyme. This particular structure is called a holoenzyme. A cofactor is a non-protein inorganic substance. It is a non-protein inorganic substance which is dialyzable. Obviously, it has to be thermostable, and the point of difference is that it is very firmly attached to the protein component. It is very firmly attached. So here are a few examples in enzymes, जैसे कि pyruvate kinase. Pyruvate kinase, you will study this in glycolysis. The cofactor, the important metal ion for enzyme activity is K plus ion. Nickel ion is there in urease. So these metal ions are essential for the functioning of these enzymes. DNA polymerase requires the magnesium ion, very important. Whereas if I draw the functional proteinaceous apoenzyme, along with a complex organic molecule, not inorganic. This time it is a complex organic molecule binded with the structure of the enzyme. This organic molecule is called a coenzyme or a prosthetic group. So you can have a prosthetic group. You can also have, like I drew in the previous slide, you could also have a metal ion along with it. Whatever the cofactor, whatever be the structure, all of this together is called a holoenzyme. The point of difference between a cofactor and a prosthetic group is that obviously the coenzyme, these are metal ions, so they are inorganic, but the prosthetic group or the coenzyme, these are larger organic molecules that are attached to the enzyme. Okay, and the cofactor, it was very firmly attached, very tightly attached to the enzyme, but the prosthetic group is loose in mint, and these are mostly derived from vitamins. Organic मतलब कहाँ से बन के आया है? It is vitamin derived. So this screen on enzymes that are derived from the vitamins, like example thiamine pyrophosphate derived from vitamin B1, and so on. Vitamin C contains the ascorbic acid, biocytin, and biotin, like that. And what is the function of of these vitamins and their deficiency diseases? With that said, I come to an end of this lecture, which discussed the structure of enzymes a little bit in detail. I hope the concepts are clear to you. If you have any suggestions or questions, queries, please do leave it down in the comment section below. Thank you. Bye bye. Have a great day.